Thank you very much. It's a pleasure to be here this evening. See lots of familiar faces, and I'd say right off the bat, I'd like to do this in a somewhat interactive way, and so if you have questions or uncertainties or ambiguities that I've raised in your minds along the way, don't hesitate to stop me. So yeah, this is what we're going to talk about tonight. We'll flit among human genetics, genomics, evolution, and talk about some medical consequences. And by the end of the latter part of the talk, we will get into some fairly gory molecular biology, but we'll try to introduce you to that in a gentle way. And so with that spirit in mind, let me begin with a 3-minute crash course in modern human molecular genetics. Okay, so this is going to be the teaching part. You got to take careful notes because there will be an exam at the end. So, coming from MIT, we like numbers, so we're going to think about some numbers that relate to our bodies and our genomes. So, first of all, what is the basic building block of life? Well, cells. Oh, let's go with cells. Okay. So let's say a cell. Now, what's this thing in the middle? This cell? Very good. Okay, right. This is a very primed audience, I can tell. Okay. How many cells do you have in your body? A lot. Okay. We seem to have consensus on this. I'm going to suggest, yeah, all right. What's outside the nucleus? The cytoplasm, right. So you've got in your body a lot of cells, let's say about 10 trillion per person. Each of you's got about 10 trillion cells, something like that. So now this is in the land of genomics. So I will ask,
Wait, does that include like hitchhikers too, or is that
these are your own cells, not the hitchhikers. If we included the hitchhikers, it would be a far greater number. If we were to talk of your metabiome, it would be a much larger number. I'm speaking of you as you are more traditionally defined, minus your intestinal contents and such. Okay. The inhabitants of your skin and your scalp and all of that. We're going to leave that all behind. For the moment, we're going to focus on the human genome. Okay, so let's then think about the components of the genome. So, how many genomes per cell? Trick question here. 1, 2. Depends on your point of view. I'm going to suggest you actually have 2 genomes in most of your cells. There are some special cells that you make, which we'll eventually get to, called sex cells, or gametes, that actually have 1 genome. So basically an egg contributes 1 genome, a sperm contributes 1 genome. At fertilization, you bring those 2 genomes together, and each of your 10 trillion cells or so contain those 2 genomes, the one contributed by each of your parents. Okay. If we go down in levels, what's the next level? There's a question.
I'm sorry, it's 2 per cell, but isn't only 1 active?
Oh, good question. You're thinking about a special chromosome called the X chromosome. We'll talk about that. We'll talk about that later, perhaps. No, both of these genomes are active.
They're both active.
Both very active.
Okay.
I think one or the other of your parents would be quite insulted by the suggestion that only one of their contributions is active. It's actually both of your parents. Right.
Thank you.
I won't ask you to suggest which of your parents you think is the active one in your own gene. All right, but that's a good suggestion. Something we could work on in the future, perhaps. But going to smaller levels below that of the genome, what's the next level of organization down? What's a subset of the genome? Chromosomes, right? Okay, next level down, chromosomes. So how many chromosomes do you have per cell? 46, that was quick. Okay, and they come in pairs, right? So we have 46 per cell, 23 pairs, because each of the gametes carries 23 chromosomes. Egg has 23, sperm has 23, you put them together, you got 46, 23 pairs. Next level of organization down below the chromosome is the gene. Very good. How many genes do you have per genome? 20,000. You guys are very modern, because if I had asked this question 15 years ago, it was 100,000. And there's been some connection between the gene count in the human genome and the equities market. And so maybe we should check again. It could be very recently on the rise, but the estimates are usually about 23,000 per genome.
When you say something is a gene, what does that mean?
Hard question. I'm going to save that for the second hour of my lecture. Okay. All right. But I will ask you, what are genes themselves? If we keep going down in levels, what is the next level of organization? Base pairs or nucleotides. So I'm going to say base pairs of DNA. And how many base pairs of DNA do you have per genome? About 3 billion, right, approximately. That's good. We've got about, oh yeah, and I say these come in two flavors. You can have A's. Basically, we got 4 letters in the alphabet of DNA: A, C, G, and T. And the pairs, an A is always paired with a T, a G with a C. So we speak of base pairs. And you've got about 3 billion per genome. And we can refer to all of these, the chemical substance, which makes up everything from the genome, chromosome, the gene, the base pairs on down. This is all DNA, right? So we could refer to human DNA at any of these levels of organization. But again, if you just keep this, this little score sheet on your refrigerator door, you will be able to read the science articles in your favorite newspaper, assuming any of you still read newspapers? Okay, right. So, right, the people who come out to lectures like this actually do read newspapers. I'm with you on this. Okay. Okay. So, and then the gene encodes a particular active sort of molecule, typically called a protein, and so if there are 23,000 genes per genome, how many different proteins, and this is a trick question, how many different proteins does a typical human cell, different kinds of human proteins does a typical human cell produce or consist of? 100,000. Well, it's a very tricky question in a way, and I will just suggest, should say that DNA, of course, resides in the nucleus of the cell. Just to complete, come back to the beginning. And proteins, we think that there are about, typically, the various cell types. And you have about 1,000 different cell types that make up your body. About 1,000 different cells that collectively comprise this 10 trillion per body. Any given cell type is expressing about 10,000 of the 23 or so thousand genes. Okay, and that's what it's subsets of those 23,000 genes are expressed in each cell type, allows those cell types to be different one from another. All right, so that is our crash course in Human Genome 101. Now, so we are now ready, yes?
And just in aggregate across all cell types, is it a one-to-one, a roughly one-to-one mapping as far as number of proteins and genes, or is protein folding?
I'm not getting into the intricacies of protein folding. I'm just saying at a very crude level, of the 1,000 or so cell types in your body, each of those 1,000 cell types is producing approximately 10,000 different proteins. Now, you can see that there must be a lot of overlap in the sets of proteins produced in different cell types, and indeed that is the case. A fair number of those proteins are absolutely essential for the viability of individual cells. So it's a smaller fraction of these 10,000 that actually specify the unique individuality of one cell type as distinct from another. Okay, that would be a whole nother, we'll go into that in more detail in the third hour this evening. Okay, all right.
He's asking over all his cells, does he have
Yes, over all your, I'm accounting for this in the sense that over all the cell types of your body, these 23,000 genes are expressed. Or actually, some of them might have just been expressed in an earlier stage, like during embryogenesis. There would be some genes that are actually active only during embryonic or fetal development. Okay, now, but we're going to get back to our subject tonight, which is human reproduction. So, human reproduction is sexual reproduction. So, how old is sex? Let's put this in a big evolutionary context. How old is sex? 400 years old. No. Okay, well, 400 million. Okay, great. So there's one. Well, let's start with some easier questions then for this audience. How old is the universe? 4.5. Save that. That is the right answer to the next question. Okay, what is the age of the universe? This is, you all have opinions. This is fantastic. Well, at least I'm glad to know that the age of the universe is in this audience expressed at least in billions of years. That's very helpful. Something like 14 billion years. Okay. So how old is the planet? The answer was 4.5 billion years. Okay. So if the planet is 4.5 billion years old, how old is sex? Well, I'm going to suggest in the evolution of sexual reproduction, there were 4 great inventions, 4 great leaps forward. And I will suggest that the first of these inventions is simply the ability to swap hereditary material. We call this sexual recombination. And I will credit, with taking sex to this level, I will credit bacteria, prokaryotes, who have various elaborate mechanisms for exchanging genetic material. And I will suggest that they invented sex on the order of 3 to 4 billion years ago. So sex is pretty old against the age of the planet, even against the age of the universe. But bacteria do sex in a very free-form, uninhibited way, compared to us eukaryotes with nuclei. Basically, when eukaryotes evolved, we institutionalized sex. And gave it certain rules. In fact, we put genes in pairs. Now, you all know that genes come in pairs. How many of you have, at some point or another, studied Mendelian genetics? The laws of Mendel. Okay. So, genes, why are genes in pairs? Well, perhaps so we can study the laws of Mendel, right? Because Mendel's about genes in pairs. And so, maybe that's what it's about. I'm going to suggest that genes are in pairs so that you can divide them in 2 at meiosis. That's the process involved in making eggs and sperm when you cut the number of chromosomes in half and you restore the full count at fertilization. And so this brings with it an alternation of, in organisms that reproduce sexually, have what we call haploid phases, where they have just 1 pair of chromosomes, and diploid phases, where the chromosomes are in pairs. Remember, we talked about most of the human cells have 23 pairs, but the sex cells have just 23 chromosomes each. And I'm putting forward as a poster species for taking sex to this level, brewer's yeast. And this is just a picture of some little microscopic picture of some yeast. And the main thing I want to point out is that we think that meiosis, that is, putting genes in pairs and the ability to divide them and bring them back together, it looks like this evolved once on our planet. Institutionalized sex evolved once, on the order of 1.5 to 2 billion years ago. So even the way we reproduce basically evolved in our eukaryotic forebears about 2 billion years ago. Now, in fact, there are a few eukaryotic organisms that do not reproduce sexually. Matt Messelson here at Harvard famously studies some of the deltoid rotifers that don't reproduce sexually. Well, I'll make the point that that is the exception, not the rule. In fact, you can make a whole career out of, and you can publish a paper whenever you find a eukaryote that doesn't reproduce sexually, because it's the exception, not the rule. But the interesting thing about these yeast though they make two different kinds of gametes, or they're actually in yeast called spores, that have one set of chromosomes. Those two types of spores are structurally indistinguishable from each other. So the next invention in the evolution of sexual reproduction was to have dimorphic gametes, to have two different kinds of gametes. And whenever one of the gametes is big, we call it the egg, and the little one we call the sperm. And whoever makes the big gamete is called a female. Whoever makes the little gamete is called the male. This is, in fact, the most broadly applicable definition of male and female. Now, don't try to use it on the street. It doesn't work all that well, but it is the most broadly biologically applicable definition. And it looks like dimorphic gametes were invented several times, quite a few times. In our own lineage, that is in animals, it looks like our forebears figured out how to make gametes of two different kinds on the order of 600 million years ago. But I'm going to put forward as a poster species for taking sexual reproduction to this level, I will put forward the turtle. Now, why the turtle? Well, turtles have males and females. The males make sperm, the females make eggs. Both sexes do meiosis to produce those gametes. But turtles have not gone on to invention number 4, which is sex chromosomes. Turtles do not have sex chromosomes. If you're a turtle, whether you develop as a male or a female is determined by the temperature at which the egg incubates. So my point here is that you can have male and female without sex chromosomes. But a fourth invention which comes along piled on top of these earlier, more fundamental inventions are the sex chromosomes, which come in various flavors. In mammals, females are XX, males XY. In birds, for example, it's the other way around. It's the male who has the 2 paired sex chromosomes, and it's the female bird that actually has 2 different sex chromosomes. Yeah. Oh, sorry. Okay. Sorry about that. Okay. Is that better? And it looks like sex chromosomes evolved independently many, many times in plant and animal evolution. Our own sex chromosomes are among the oldest known sex chromosomes. That is, in mammals, our sex chromosomes began to evolve from ordinary autosomes, ordinary matched chromosomes, on the order of 200 to 300 million years ago. And what I'd like to do now is I'm actually interested deeply in all of these levels, and we might in the 4th and 5th hours of the evening turn to them. But since I know some of you have to leave after the first hour, I'd like to turn to the sex chromosomes. So, truth be known, I have spent the better part of the last 25 years defending the honor of this small downtrodden chromosome in the face of innumerable insults to its character and its future prospects. And so it is with that spirit in mind that I'd like to present to you some observations on the evolution of the sex chromosomes, and we'll see where it goes medically from there. So this is actually an electron micrograph of, on the left, the stately and robust human X chromosome, and it's rather, it's appropriate here. The Y chromosome sort of has its head down. It's a rather demure, even shy fellow here on the right. But let's see what we have to say about. Let me give you a little bit of an intellectual history of thinking about the Y chromosome. Mendel was working in that pea garden in the 1860s. In the first few decades after he made some discoveries, not much heed was paid. But in the earliest years of the 20th century, Mendels Rules were rediscovered and very quickly applied to to our species. So in the first decade of the 20th century, people were looking around and defined the 3 big modes of inheritance that you've all studied in your first biology course. Defined the X-linked recessive mode of inheritance, autosomal recessive, and autosomal dominant. But also in that decade, people were looking for traits that might be inherited according to a 4th mode of inheritance, the Y-linked mode. The search was on for genes on the Y chromosome. Here was one of the early reports of success. So you may be used to looking at these diagrams of human pedigrees. You familiar with this? This means, so this is dad, males are the boxes, females are the circles, and when the thing is filled in, it means displays the trait in question. In this case, the trait in question was hairy earlobes. This was a report from 1907 in the archives of psychiatry, neuropathology, anthropology, criminology, medicine, and law. This was one of the early interdisciplinary journals. And you'll see that down in this generation, there were a couple of problems. Maybe these guys shaved their ears or something. But in any case, this was one of the early reports of success in finding a trait which appeared to be transmitted through human families as if it was passed in a clean way from father to son and on and on and on. Well, it turns out first half of the 20th century was an exciting time on the Y chromosome because not only hairy earlobes, but many other traits were ascribed to the Y chromosome. This all came crashing down in the mid-1950s. So in 1957, the president of the American Society of Human Genetics was a noted Drosophila geneticist named Kurt Stern from Berkeley. And at the annual meeting of the American Society of Human Genetics, which that year was held in Ann Arbor, Michigan, he delivered, as is the custom of the president of the American Society of Human Genetics, he delivered the presidential address. His presentation was entitled Hairy Ears, Porcupine Skin, and Other Alleged Sins of the Y Chromosome. Now, when the text of his speech was published in the Society's journal, it was cleaned up a little bit. But basically, what Stern did in this lecture was to, one by one, debunk each of the 17 presumably or possibly Y-linked traits that had accumulated during the previous 50 years. And so by the end of his presidential address, in front of all of the assembled human geneticists of North America, by the end of his presidential address, there were no genes left standing on the Y chromosome. This was, in some sense, a low point for the chromosome. Now, I'm not attacking, he was on the mark. These were just 17 examples of wishful thinking and pretty shoddy scholarship, to be honest. So he was doing the right thing. Now, 2 years later, 1959, Klinefelter's males were shown to be XXY. Turner syndrome females, XO. That demonstrated that it was the presence or absence of the Y that determined whether an individual developed as a male or a female. And that implied the existence of at least one sex-determining gene on the Y chromosome. So by 1959, 2 years later, we knew there was one gene on the Y chromosome, but that was universally viewed as an exception on an otherwise barren genetic wasteland. And so emerged the understanding of the Y chromosome that has prevailed for most of the last half century. And there was an evolutionary perspective from which to understand the paucity of genes in the Y chromosome. So 300 million years ago, when we were reptiles, and you remember, when you get together at Thanksgiving and things, you talk about family traditions. I hope you go back that far. 300 million years ago, when we were reptiles, we had no sex chromosomes. We had only ordinary autosomes. We probably had temperature-dependent sex determination as in turtles today. But something began to happen 200 to 300 million years ago. A mutation on an ancestral autosomal pair, completely ordinary, unsuspecting pair of autosomes, a mutation began to give rise to what would become a tyrannical male-determining gene on what would become the Y chromosome. This gene lives on today as SRY. And what happened was, first in the immediate vicinity of SRY, and then in a larger region over time, what was becoming the Y and what was becoming the X stopped swapping genes at meiosis. Okay, because at meiosis, not only do you divide the number of chromosomes in half, but you actually do some trading between mom and dad before you divide the chromosomes out. And this process continued, and it turns out the ability to swap genes is really good for the long-term health and well-being of genes over evolutionary time. So what happened is that the Y chromosome, what was becoming the Y chromosome, was now isolated. It was now isolated. It no longer had a partner with which it could swap parts. So genes on the Y began to rot and decay and disappear altogether. Now, as we understand it, the X chromosome essentially retains the gene content. Today's X chromosome retains the genes of the ancestral autosome, whereas the Y, with the exception of a sex-determining gene, is a sort of shriveled remnant of its former glory. Now, if you take this idea to its logical extreme, it doesn't look good for the Y chromosome. And in fact, in an influential editorial published in an obscure scientific journal called Nature in 2002, this was all taken to its logical conclusion. So as I just told you, in this editorial said in a more scholarly way what I've just sort of mumbled, that is the Y chromosome is particularly vulnerable because it doesn't have a partner with which it can swap genes. It can't swap genes like other chromosomes do. So it can't retrieve damaged, it can't repair damaged genes by recombination, by swapping. And then came the punchline in this editorial. At the present rate of decay, the Y chromosome will self-destruct in around 10 million years. Now, we'd been working in our lab on the Y chromosome for, I don't know, almost 18 years by then. And so, fortunately, I was not the first member of my lab to read this editorial. It was actually one of my graduate students. He came running into my office, tears streaming down his face. We held an emergency lab meeting, and we resolved to pick up the pace of our research. But we weren't the only ones who were reading this. I was going through an airport lounge in the UK, and here, New Scientist is a magazine in the UK that presents science to a broad audience. And the cover story here explained that not only was the Y chromosome going to disappear, but it was going to take our species with it. And in this article, the time scale was moved up from 10 million years to 5 million years. So things were getting really pretty bad. And this has inspired other art forms. So it turns out there is a whole comic book series which is called The Last Man. And you may notice this is one of the covers. You may notice the subtle Y chromosome imagery here. And actually, this has been turned into not only a series of comic books, but a very, very bad movie, which has become a cult favorite in our laboratory. We watch it at least once a year. I'll give you a little bit of the plot of this movie. Feeling that they were better off without males, the women of Earth decided to outlaw men because they were too violent. And so they developed a weapon called the Y bomb, which resulted in the deaths of 97% of men. Then a graduate student named Hope Chase conducts a cloning experiment to produce a new male whom she names Adam. And anyway, it doesn't necessarily turn out too well. When Adam reaches maturity, he finds himself on the run. He's hiding out with rebel bands of the last remaining men. If you ever have a chance to see this movie, and I suspect and hope that none of you do, it turns out most of the last remaining men were hanging out in abandoned NFL football stadia. And occasionally the women come in on helicopters and strafe the field. Anyway. I think it might be The Last Man. It's just so embedded in our minds we don't even have to think about the. Yeah. So I tell you, no other chromosome has to put up with this. But this is not the least of it. If you do a Google search on the Y chromosome, what you'll mostly come up with are models of the Y chromosome's gene content that look something like this. So now come on, this is I'm giving a serious scientific presentation here tonight. The channel flipping gene, sometimes located here, then it hops down here. The BOLS2 gene confers self-confidence on link to ability. DC10, this has to do with the ability to identify aircraft at long distances away in the sky. Mom for you, presentation of spiders and snakes to eager mothers. The well-known Patui gene. And then something that my wife has been trying to explain to me for years. That maybe there's a claim of linkage to the difficulty in remembering anniversaries and birth dates. It's the gene down here. Anyway, no other chromosome has to put up with this, I swear. So something had to be done in the face of all these insults. So what I want to do is tell you that what we've found in recent years through a variety of genomic studies is that the Y chromosome actually does do something. It has genes. And for the most part, it's specialized in one thing, in sperm production. And this has been revealed by sequencing the chromosome, by cataloging the genes that are found on the chromosome, and by the fact that, medically speaking, deletions on the Y chromosome are among the most common causes of male infertility. So let me tell you a little bit about that. So, okay, now we're going to take it up a little notch in. So, a crash course in what we know about the Y chromosome based on genomic analyses from the last 10 or 15 years. So, anybody know what this P and Q are about? Those are the arms. Those are shorthand names for the arms. Human chromosomes have a short arm and a long arm. And why is the short arm called the P arm? Because in French, petit. And why is the long arm called Q? Because it's the next letter in the alphabet. Okay, so I'm not kidding. This is right. Okay, so so, and this thing in the middle, which breaks them, this is called the centromere, at the sort of the division, the point of division between the short arm and the long arm. Okay, now it turns out the Y chromosome differs out at the ends. These little green bits, this is very fancy information now. These little green bits are the so-called pseudo-autosomal regions of the Y chromosome. This is for you who want to, this is information for those of you who want to score really extra points at that next cocktail hour. Just drop pseudoautosomal region into conversation somehow. These are regions where the X and the Y chromosomes actually do swap genes and trade parts during sperm production, during meiosis in the male. But we're going to talk mostly about what's in between, where the Y differs from all other chromosomes in our nuclei in two respects. First, it's the only chromosome that is specific to one sex, not found in both. And it's the only chromosome that doesn't cross over. It's another word for this swapping of genes that occurs at meiosis. It's the only part of our nuclear genome that doesn't regularly participate in swapping or crossing over with another chromosome. Now, it turns out that much of the long arm of the Y chromosome, yes?
The pseudoautosomal is rarely swapped.
In the pseudoautosomal regions, there is always swapping in the production of sperm.
Okay, so I'm just confused.
Yeah,
So that always swaps over, but with another chromosome.
with the X.
Okay.
Okay? So out of these tips, and we're going to actually come back to a little bit of a movie of this. Out of the tips, the X and the Y chromosomes actually swap parts. But in the middle, in the great bulk of the middle of the Y chromosome, no trading of parts with the X.
If there's no crossing over
Yeah,
for the Y,
except at those tips.
but is there any crossing over in the X?
Yes. Okay, good question. So the X is not crossing over apart from these pseudoautosomal tips in male meiosis, in the making of sperm. But think about what's happening in the making of eggs. You have two X's, and they trade parts, just like other chromosomes. Okay? So that's how the X stays healthy over evolutionary time, because it's involved in swapping, but in female meiosis, not in male meiosis. Okay? So, turns out much of the long arm of the Y chromosome has a very simple monotonous sequence composition, and we refer to it as heterochromatin. Now, sadly, this sequence composition over here is so monotonous and confusing that no molecular geneticist has entered the heterochromatic region and returned alive. So we're not going to speak of it anymore tonight. We'll instead move on to the euchromatic part of the Y chromosome, which is 23 megabases, or 23 million base pairs, or about a little less than 1% of the human genome. And within that region, I'll just now give you a really quick crash course in what have we learned about the Y chromosome by sequencing it. Now I'm going to contrast old and new understandings of the human Y chromosome. So I told you before that the prevailing idea was the Y was a genetic wasteland. We now know that it carries about 76 protein coding genes. They encode 27 distinct proteins. Now, why is this number of proteins less than the number of protein coding genes? It turns out a bunch of the genes on the Y chromosome, as I'll show you, are present in multiple virtually identical copies. And there's a distinct specialization of these genes in sperm production, which is to say many of the genes on the Y chromosome are only expressed in one organ, in the testis, in the cells on their way to becoming sperm. Nowhere else in the body. Very specialized. Old idea was that the Y was merely a rotting copy of an ancient autosome. What we now know, and I won't go into this in any detail, it turns out that the Y chromosome, though it has lost many genes along the way, has actually gained some fresh reinforcements, especially during primate evolution. There have been some other genes that have hopped into the Y chromosome. Believe it or not, some other genes have decided to take up residence in this relatively inhospitable territory. And some of those genes have actually been amplified once they arrived on the Y chromosome. Full of junky repeats. That's what I was told for the first 15 years that I worked on the Y chromosome. What I'm going to show you is that the Y chromosome is actually structurally spectacular. It contains gene-rich palindromes of enormous scale and precision. It was the prevailing view was that in the absence of crossing over, gene swapping of the kind that we were familiar with, it must be the case that all genes in the Y were disintegrating. But what we've actually come to understand, and I'll talk about in the next minutes, is that a different kind of recombination called gene conversion might allow for the maintenance of genes on the Y chromosome over evolutionary time. And finally, the notion that the Y is headed for extinction, from what we found is that even the genes that are found only once on the Y and that cannot partake of this very special mechanism of swapping, even the genes that are found only once in the Y seem to be preserved by the process of natural selection to a degree that the theorists had not given the Y full credit for. And some of this was reinforced by our comparative sequencing, which I'll not say too much more about. Just a month or two ago, we published the sequence of a second Y chromosome. That was the chimpanzee Y chromosome. And the comparison of the human and chimp Y chromosomes really clearly reinforced this idea that natural selection is working better on the Y than we had suspected. So anyway, it's a quick summary of what we've learned. And I'm going to talk in particular now about two aspects of the Y chromosome, these palindromes, and special mechanisms that they feature. So when we published the sequence of the human Y chromosome, the subtitle was A Genetic Hall of Mirrors. And what I want to do is now take you into the genetic hall of mirrors that is the Y chromosome. The Y chromosome that we know today as opposed to the genetic wasteland of 50 years ago. So what is a palindrome? You guys, what's a palindrome? It's something that reads the same forwards and back. So there are plenty of, you maybe know some palindromic statements, or one that's relevant in this case is, Madam, I'm Adam. But there are others. But the ones on the Y are much more spectacular than any palindrome you've ever encountered before. So I'll give you a sort of a structure of a human Y chromosome palindrome. So first, so these are sequences of A's and C's and G's and T's that run in this direction and then in the opposite direction. And they show almost perfect left arm to right arm symmetry. These are the degrees of identity that we observe between the left and the right arms. And if you see how many decimal places there are here, that means they must be big. And they are. I should say, I'll flip this around and say, this means that the divergence between the left and the right arms is less than 0.06%. So they're virtually identical in sequence. And they are very big. So the largest of the palindromes in the human Y chromosome has an arm length of almost 1.5 million base pairs of DNA. That is to say, a wingspan of 3 million base pairs of DNA. That one palindrome actually constitutes 1,000th of the human genome. So imagine if there were a perfectly symmetrical structure that comprised 1,000th of the landmass of North America. People would go to see it. In fact, you'd probably build you'd declare it to be a national park. And actually, I've often wondered why, maybe this is why so many of our most famous national parks begin with the letter Y. Yosemite, Yellowstone. You see what happens to the brain if you work on something sort of too long. Anyway, now these are not just pretty structures. They actually carry, I should say, in the middle there's a non-duplicated spacer, but those tend to be quite small compared to the massive arms. And lurking on the arms of these palindromes are genes that are expressed in the testis in the production of sperm. So, and when we began to realize this some years ago, it was a sort of a mind-boggling, transforming realization for us. Because, so what it means is you've got a gene that is, let's say, running in this direction, and then you have a mirror image copy of that gene that is running in the opposite direction. So the one thing, prior to this discovery, the one thing that we knew was true about the Y that distinguished it from all other chromosomes, the one thing we knew was true was that genes on the Y did not come in pairs. But now, all of a sudden, we realized, oh, genes on the Y come in pairs. It's just not one on the chromosome you got from mom and one on the chromosome you got from dad. The pairs are built into the arms of a palindrome. And it turns out this very slight degree of arm-to-arm divergence in the Y palindromes, it required explanation. What could account for this? Well, there are two possible explanations. Either these palindromes were of very, very recent origin, and the molecular clock people would say they had to be less than 500,000 years old, or there was some ongoing mechanism of homogenizing the sequence, of maintaining identity between the sequence. And this would, it turns out, if you just swapped the sequences between the left and the right arms, they could still diverge. You wouldn't homogenize them. So it had to be a mechanism of overwriting the sequence from one arm onto the other periodically. And this would be non-reciprocal recombination, what the geneticists call gene conversion, where instead of swapping, you actually replace the one, you overwrite. And operationally, this came down to a simple prediction. If it's gene conversion, then you should find the same, you might find the same palindromes in our closest relative, living relative, the chimpanzee. But if they're of recent origin, you certainly shouldn't find them in the chimp. Well, it turns out we found them in the chimp. Found chimp counterparts of the human Y palindromes. And so I'll just show you here. So here's, let's say, a simple diagram of a palindrome on the chimpanzee Y chromosome, showing a very slight degree of arm-to-arm divergence. And here is the same palindrome. This is one of my colleagues at Whitehead. To this point, I thought, can you guys keep a secret? Because I never actually, I'm not supposed to publicly disclose whose Y chromosome we sequenced. But anyway, you look like a suitable donor. So anyway, Bob Weinberg over here also has a very, oh, did I mention his? Okay. Has a very, again, a very slight degree of arm-to-arm divergence between the arms of his palindrome. But thankfully, and Bob is very pleased with this, there is some degree of divergence between himself and the chimp. Okay, now it's not a lot, but it's 1.44%. So the point is, these two arms are virtually identical. These two arms are virtually identical, but there's this degree of difference between the two. This implies a couple of things. First of all, it implies that the palindrome existed in the common ancestor of human and chimp 6 or 7 million years ago. It's the first inference. Second is, it implies that there has been, along this lineage, the chimp lineage, and along the human lineage, there has been ongoing homogenization. So that you can end up with two copies virtually identical, but some divergence between the pairs. Okay. So, and it turns out these palindromes comprise, there's not just one, there's a bunch of them. And they make up collectively about 25% of the euchromatin, the business part of the human Y chromosomes. Here's the biggest one I showed you. Here are the others. And they carry all of the intact copies of the long arms spermatogenesis genes. So the long arm of the Y chromosome is about spermatogenesis. Those genes are found on palindromes, which is to say, so, and here are these 1, 2, 3, 4, 5, 6, let me see, 1, 2, 3, 4, 5, 6, 7, 8 different gene families that are found on the long arm of the Y chromosome and where their members are located. For instance, the DAS genes down on the bottom, There are 4 of them on most human Y chromosomes. There are 2 in a sort of palindromic couplet here, and here's another palindromic couplet. So all these genes exist exclusively as pairs, as mirror image pairs. Yes?
Are there palindromes on other chromosomes, or is it?
Are there palindromes on other chromosomes? Well, it turns out,
He wants to know if she has any.
are you feeling left out of this story in some way?
I don't know how significant some of these other,
Will it be more significant to you if there are? Well, yeah, it turns out there are, it looks like the greatest concentration of palindromes in the human genome is on the Y. It looks like the second greatest concentration, and it makes up a much smaller fraction. It looks like the second greatest concentration may be on the X chromosome. But it turns out that though we all know the human genome has been sequenced, it turns out it has to be sequenced to really, really exacting standards to even be able to see these palindromes. Okay? And so I would actually say, and this might be controversial to some ears, I don't think the human genome has yet been sequenced with sufficient accuracy to fully answer your question. Okay. There are secrets yet to be uncovered in our sequenced genome, right? Okay, so, yes, yes.
I just wondered if you could tell if somebody's a homosexual by looking at their genome.
Can you tell anything about sexual preference by looking at the genome? No. Now, the question, in a sense, your question begs another question, which is, what do we know about the genetic basis of sexual preference? There are any number of aspects of sexuality that where one might explore the genetic basis. And to be honest, there's not a lot of work ongoing today. There's not much work ongoing today into the genetic basis of sexual preference. There were some discoveries made almost 2 decades ago now, which suggested that there were some genes which were connected to matters of sexual preference. But this is not a worked out question by any means, and there is not much work ongoing at present. It's not easy to get funding, as it turns out, to pursue this question. And we could discuss the politics of that at great length. Maybe the 6th hour we'll go there. Was there another question in the back? Yeah. Dominance in monkey troops. Ah, right. Okay. So, great question. We'll take that up in the 8th hour tonight. So we're actually very, very interested in the question of how differences in mating behavior may affect the evolution of genomes, including the evolution of the Y chromosome. And when we reported just a couple months ago the sequence of the chimpanzee Y chromosome, we were amazed by the degree of difference between the human Y and the chimp Y. And in searching for possible explanations, we reached out speculatively to point out the differences in mating behaviors of our own species and chimpanzees. And there's probably much more to be learned in this space. But you're asked, these are all great questions. Yeah, we're going to have to take them up in some extended session here, I think. So, having shown you just the outline here, I'm going to show you now just a little bit of a movie to try to connect. Well, let me just, if you could just hold it, because the movie's going to want to run on its timing, not mine or yours. So let's see if we can. So here is an overview of the human chromosomes. You've seen pictures like this. Yeah, karyotype, right. So here we're going to focus in on chromosome 5, an ordinary pair of chromosomes. You get one from mom, one from dad. Crossing over or swapping can occur anywhere between the chromosomes you got from mom and those you got from dad. A little kiss and swap up here. We'll do a little something similar down below. So you see, before the chromosomes get divided, they can trade parts like that. That's what happens on ordinary chromosomes. But now we're going to go in and look at the sex chromosomes, so the rules of the game are different. So now we're going to go to the X and Y. Remember I told you about the pseudoautosomal regions. Out at the tips, the X and the Y act like ordinary chromosomes. Okay? Out at the tips, they act like ordinary chromosomes. They sort of take a look at each other. A little kiss and swap can occur up here. So you regularly, this is in the making of sperm, right? This is the making of sperm. Regularly exchange bits. The same can happen down here. But now we're going to look at the innards of the Y chromosome, which doesn't participate in exchange with the X. But instead, we're now going to blow this up, and we're going to make a bit of a palindrome in there and show you what I mean by non-reciprocal recombination. So imagine we've got this palindrome like so, and we've given it a big spacer for purposes of the illustration here. What we imagine is that this, so what if a base pair substitution, what if a mutation occurs on one arm of that palindrome? What we imagine is that, that it's possible for these two arms to pair, and now in a non-reciprocal fashion, for the intact copy to overwrite this and repair it. Now, looks good to you, right? This is sort of gene therapy, right? But is this really a weighted coin? How does it know to fix the bad mutation with the original good? It probably doesn't. It's probably not weighted. It can go probably in either way. Gene swapping is not necessarily always going to go for the good. So let's replay this now. And we will have, now in this case, we're going to have the mutation overwrite. Okay? So presto, like so. The point is that by either of these means, the result is to maintain the virtual identity of sequence of the left and the right arms. So we think it's a non-reciprocal recombination mechanism of this sort. And, okay, so I am at a transition point. I see that it is 7:05. And I have a lot more material that I could present. But maybe...
I think since we've incorporated questions, we should go at least, 7:15 and see where we are.
Okay, because I haven't gotten really to the medical part yet. So let me give a little bit of medicine here then. Okay, so what I want to suggest then, and we're going to fly now. So I'm going to deflect any extraneous questions at this point till into the 9th hour. So, okay. So now that we've got this, I want to pose a question. What if the whole chromosome turned into a palindrome? And what we've found over the last 15 years is the most common genetic cause of spermatogenic failure are deletions in the Y chromosome. So missing some of those palindromes is the most common genetic cause of spermatogenic failure in our species. And I'm going to give you a quick summary. This is now you can get, if anybody wants to get continuing medical education credits, this would be a good point to sign up. So I'm going to give you a quick summary of what we've learned in the last 15 years about Y chromosome deletions and male infertility. So it turns out I'm going to refer to interstitial deletions, when a middle part of the Y chromosome is missing. So it turns out there's one deletion of the Y chromosome. The yellow bar shows a piece that's missing in some men with spermatogenic failure. That deletion is reasonably common. It actually occurs, we think, in about 1 in every 2,500 newborn boys. It's missing that piece of the Y chromosome. And there are 4 less frequent deletions, whose extents are shown like so. These deletions are big. The smallest of these takes out 800,000 base pairs. The biggest of these deletions takes out almost 8 million base pairs of DNA. What is most remarkable is that men missing those 8 million base pairs of DNA, first, what's most remarkable is that they're alive. Any other part of the genome, if you're missing 8 million base pairs of DNA, guess what? You don't make it to birth. But these men, not only are they alive, they only have to go see one physician. They're only going to show up for their infertility doc. They don't have any other medical problems. It speaks to the amazing functional specialization of the long arm of the Y chromosome. And, I should say, the men who have spermatogenic failure and are missing a part of the Y chromosome, from whom did they inherit their Y chromosomes? Their fathers. Were their fathers fertile? In all likelihood, their fathers were fertile. So, in most cases, these are new deletions. This is a funny kind of genetics. You know, you're used to the kind of genetics. What you got taught in your biology, in your first biology course, was traits that could be passed from one generation to the next. So these are, in these cases, the fathers have intact Y chromosomes. The son inherits a Y chromosome with a deletion and is unable to pass it on. So this is the genetics of new mutations. And these are recurrent. So it turns out that palindromic hall of mirrors structure of the Y chromosome is a fragile one. It is susceptible to these deletions. And so, in fact, this AZFC region, this deletion like so, in our laboratory, we have studied 87 unrelated men who all have essentially the same deletion. In other words, lightning has struck at the same 2 points on their Y chromosome because there's a particularly fragile sites there, in a sense, because of this palindromic structure. And it turns out these delete many genes. In the case of the biggest deletions, up to 25 different genes involved in sperm production. So this was basically what we knew up until a couple of years ago. There was a lot of interest in these interstitial deletions causing spermatogenic failure. But then there was some other observations that have been piling up over the years that didn't quite fit this picture. In addition to men who were missing an internal part of the Y chromosome with sort of 2 breakpoints, there were other men with spermatogenic failure who had deletions with only 1 breakpoint, like so. They seem to be missing everything from 1 point to the end of the chromosome. So they seem to be missing a much bigger part. And are these truly terminal deletions? Well, for those of you who know about chromosome biology and have heard about things like telomeres and such, you know that a chromosome kind of has to have a cap on the end. In fact, there was a very recent Nobel Prize for the understanding of telomerase. And so we really wondered, are these truly missing the cap on the end of the chromosome? And then another curious thing, it looked like these deletions most of these breakpoints were falling within palindromes. They seemed to correspond to the locations of the palindromes. And this got us thinking a crazy, crazy idea. That what if you could turn the whole chromosome into a palindrome via a little variation on that mechanism of gene conversion that I told you about before? What if you occasionally had the Y chromosome do something like so? It turns out in the production of sperm, when you go into meiosis, the first thing that happens is that all the chromosomes get doubled. So you go from having a pair of each chromosome to having 4. And even the Y gets doubled. What we wondered was, what if occasionally 2 copies of the Y chromosome found themselves in a cell, and what if they occasionally got misoriented like so? And what if you had an actual crossover at one of these mirror image sites. You know, a mirror image site would be as good a target for crossing over in either orientation. Admittedly, a crazy idea, but the result could be the production of a mirror image chromosome. Okay, so now, is there any truth to this? Well, it turned out this would yield a mirror image Y, what would be called an isodicentric chromosome. Iso simply means symmetric, same in both directions. Dicentric would mean carrying 2 centromeres. So a normal Y has 1 centromere. This thing would have 2 centromeres. That was the proposition. And it turned out that this was the case. So here I'll just show you some fluorescence in situ hybridization images, use some DNA probes to actually look at these chromosomes, at real chromosomes. So we're going to use a DNA probe for a spot out here, and this probe is going to be red, and a spot for the centromere in green. And this is what it looks like on a normal Y chromosome. We've got the green spots depicting the centromere, these red spots depicting this point. Here was one of these mirror image chromosomes, which instead of just going red-green, went red-green, green-red. And they came in different sizes. So some were bigger. And it turns out that with these preliminary observations in mind, we went back over all of the patient DNA samples that our lab had studied over the last 25 years or so. And these were individuals who we had studied in our lab for any one of 3 reasons. A discordance between the sexual anatomy and the sex chromosome constitution. That is, you expect male anatomy to match up with a Y chromosome, female anatomy to match up with sort of an XX sex chromosome constitution. These were cases where there appeared to be a mismatch. And we also included individuals who we had studied over the years who had a microscopically detectable, by light microscopy, an anomaly of the Y chromosome. And we also studied men with little or no sperm production who didn't meet either of these 2 criteria. And out of those over 2,000 individuals, we found actually 60 people who carried these mirror image Y chromosomes. And 51 of these arose through this mechanism of crossing over at the palindromes that I depicted for you in cartoon fashion. And 8 different palindromes on the Y were targets of these events, suggesting that this is a fairly general phenomenon across the Y. And it turns out this is a significant cause of spermatogenic failure in our species. Now, I'll close with an interesting tidbit which concerns how we come to develop male or female anatomy as embryos and fetuses. It turns out that of these 60 individuals in whom we found these mirror image Y chromosomes, 20 of these individuals were anatomically feminized. Now, what do I mean by this? Well, when a baby is born, what is the first question asked? Is it a boy or a girl? How do you answer that question?
Does he have a penis, I guess?
You take a look between the legs. This is a scientific assay, right? And so it turns out that of these 60 individuals in whom we'd found the mirror image Y chromosomes, 20 either had fully feminized external genitalia or at least partially feminized external genitalia. 20 out of 60 was pretty remarkable. And so how could that be? Well, One possible explanation would be that the SRY sex-determining gene had gotten lost in the process of making this mirror image chromosome. That would be a very straightforward explanation. And actually, that was true in 2 of these individuals. The SRY gene was not present on the mirror image chromosome. So that left only 18 to explain. So 18 of these individuals had SRY present not just once, but on both ends of the mirror image chromosome. Okay, so now we'll try to close with the solution to this puzzle. We came up with another speculation. That is that maybe the answer lies in the fact that these mirror image chromosomes have 2 centromeres. Now it turns out that centromeres play a very important role in making sure that whenever a cell divides to become 2 cells, that each of those daughter cells receives a copy of each of the chromosomes. It's the centromere plays a critical role in parsing out one copy of each chromosome to each of the daughter cells. So we wondered if chromosomes that had 2 centromeres might get confused in this process of divvying up the chromosome copies. And we thought that maybe this dicentric chromosome would sometimes get lost in the shuffle because it had 2 centromeres and the mechanism of parsing the chromosomes would get confused. Maybe the result would occasionally be a so-called XO cell that had 1 X chromosome, had completely lost the mirror image Y. And if this occurred during early embryogenesis, if you had XO cells in the embryonic gonad, the structure which will give rise to either the testis or the ovary, it's going to be as in a Turner embryo, and the result will be female development, okay, including feminization of the external genitalia. And it turns out that there was a testable prediction. We scientists really like testable predictions. We're kind of predictable ourselves that way. We like testable predictions. And it led to a specific prediction. It turns out that geneticists studying yeast chromosomes with 2 centromeres had discovered more than 2 decades ago that the instability, the confusion in cell division is greater when the distance between the 2 centromeres is greater. Okay, so if you have the 2 centromeres kind of close together, cell might know what to do with them. Put them far apart, cell might get really confused. So the prediction was the greater the distance between the centromeres, the higher the probability of anatomic feminization. In other words, the greater the distance between the centromeres, the greater the chance of loss of that mirror image Y, and the greater the chance of feminization. And so we're just going to look then for a correlation between it turns out, the size of the mirror image chromosome and the likelihood of male or female development. So, and it turns out we found the correlation we're looking for. Now, most of the time we make predictions and they're all wrong. This was one of these rare instances where the prediction held. So I'm just going to show you, these were the targeted palindromes in these events. And stay with me. The mirror image chromosome starts at the end of the short arm over here. Starts at the end of the short arm over here, comes through the centromere, goes to the targeted palindrome, and back out the other side. Okay? So it turns out the more distal the targeted palindrome on the long arm of the Y, the greater the distance between the 2 centromeres, the bigger the mirror image chromosome. So our prediction was that the females would be shifted, their targeted breakpoints would be shifted to the right, the males would be shifted to the left. And this is what we saw. Here were the anatomically male individuals, 40, and each of these dots represents one individual, the site of reflection of their mirror image chromosome. Again, starting over here, reflecting at this point, going back out. Each of these represents one individual. And this was the distribution in the anatomically feminized individuals. So a shift to the right in the females, a shift to the left in the males. And so it actually turns out that the average distance between the centromeres in the females was twice that in the males. So ironically, in human beings with mirror image Y chromosomes, the more Y DNA you have, the greater the likelihood you're a female, because that Y chromosome is unstable. And so I'll just close with some wild speculations that arise from this. So could the mitotic instability of these mirror image chromosomes be a significant cause of the XO condition that we know as Turner syndrome in girls and women? So I'm telling you about cases in which we could detect the mirror image chromosome. What if that chromosome were so unstable that even though it might have been present in the fertilized egg, that it was lost entirely in the body? You would call that individual a Turner syndrome individual. But they might have originally had the mirror image Y chromosome. Now you'd say, this is crazy. This is even crazier than the whole idea that you'd make these mirror image Ys. Well, there are a couple of facts about Turner syndrome that are relevant here. So you might think, what do you know about, let's think about another situation in our species where the chromosome number is affected. How about trisomy 21? So we know there are a couple of things that are not true in Turner syndrome. So it turns out, while there is a maternal age effect, while mother's age is a risk factor with regard to trisomy 21, there is no maternal age effect for Turner syndrome. In other words, the mothers of girls with Turner syndrome are not older on average than the mothers of girls without Turner syndrome. And why is this relevant? Because we think that trisomy 21 arises from a process that is pre-fertilization. It's during the making of eggs in the mother. But I'm suggesting that Turner syndrome might be arising through something that occurs after fertilization. It might be unrelated and have no relation whatsoever to anything going on in the mother. And it turns out that if we ask in girls and women with Turner syndrome, that single X chromosome, we can ask using genetic markers, is the X chromosome that's retained, does it come from mom or does it come from dad? And it turns out, in 3/4 of XO girls and women, the X chromosome, the single X chromosome, is from mom. Okay? So mom's off the hook. It's dad's sex, it's a sex chromosome from dad that's missing. And since these are girls, you'd automatically think, oh, it's dad's X that's missing. But it could just as easily be dad's Y that's missing, or a derivative of dad's Y that's missing. And so it's just a wild speculation that Y palindrome crossing over could, ironically, be a cause of a chromosome disorder that exclusively affects girls and women. So with that wild speculation, I'd like to thank you for your attention. You've been very generous. Thank you.